So I will show you today some different types of biological networks. So I will take the help of book of Newman. So about basic properties already we have discussed of the networks. So you can see here, yeah. So we have already discussed the basic properties of the network, and you can measure them, measure all other properties. Now here our focus is on the biological network, not on the other type of network. Okay? So for that, yeah, this is the biological network. So if you see the biological network, yeah. So in biological network, what happens? They are different types of biological networks, and you have first one is the metabolic network. The principal types of biological networks are metabolic networks, protein-protein interaction network, and gene regulatory network. So if you see about a metabolic network, it is a network of chemical processes which where the cells break down the food and nutrients using into the usable building blocks like catabolic metabolism, and then reassemble these blocks to form a biological molecules of cells. So anabolic and catabolic both are involved here, and these breakdown and reassembly process can be represented in pathways in the metabolic network. Now, generally, the vertices of the metabolic networks, they are the chemicals produced and consumed by the reactions. So these are the metabolites. So if you want to define a metabolite, it's a small molecule like carbohydrate or lipid, as well as amino acid and nucleotides, and they are the part of the this metabolic network. So, although the fundamental property of metabolism is to turn food into useful biomolecule, we have an assembly line, and metabolism is a part of not just network of conveyor belt in one reaction to another. The final product falls out within. It's a dynamic process where the concentration of metabolites can change widely and rapidly. So concentration of the metabolites is changing in the cell, and cell have a mechanism to turn on and turn off the production of particular metabolites in the cell. And so we are more focusing on metabolism because individual chemical reaction in the cell is more and more metabolite that are broken down and combined to produce one or more others. And metabolites which are consumed are known as substrate, while those who are produced are called products. Now, most of the metabolic reactions, they are not spontaneous, or even if so, they are at very low rate. Therefore, to make the reaction occur at a usable rate, a cell employs an array of chemical catalysts known as enzymes. Now, these enzymes are macromolecules, usually proteins, and occasionally RNAs. And like enzymes are not consumed in the reaction, they catalyze, but they play important role in the metabolism of the cell. They not only enable reaction that would otherwise be thermodynamic disfavored or too slow to be useful, they also provide one of the mechanism by which cell control the metabolism. So you can increase and decrease the concentration of enzyme and then catalyze a particular reaction, and also cell can turn on or turn off or moderate the speed of. So enzymes are generally highly specific for the reaction they catalyze, and they also enable one or small number of reactions, and therefore thousands of enzymes are known and many more are waiting to be discovered. And this highly specific catalysis allows a fine degree of control over the processes in the cell. Now the metabolic networks details vary between different species of organisms, but amongst all animals, at least large part are common to all and most species. Therefore, the important pathways and other subportion of the metabolic networks, they are essentially unchanged across the entire animal kingdom, and for this reason, one often refers simply to metabolism without specifying a particular species of interest. The correct representation of metabolic network is a bipartite network, and we encountered bipartite networks at different places. In social networks also we have seen. So here you can connect the metabolic network. They represent the metabolites and metabolic reactions, with agents joining each metabolite to reactions in which it participates. In fact, a metabolic network is really directed bipartite network, since some metabolites go into the reaction and some come out of the reaction. So they are the products. This bipartite representation of metabolic network not include any way of representing enzyme, though not metabolites themselves are still an important part of metabolism. Although it's not often done, one can in principle incorporate enzymes by introducing a third class of vertex that is called with edges connected to them to reaction they catalyze. And the enzymes are not consumed in the reaction, therefore the edges are undirected, running neither into or nor out of the reaction they participate. So these are the basic fundamentals of metabolic networks. And experimental part of the metabolic network is complex and laborious process, and they are probed using different radioactive isotopes to trace the intermediate products along the pathway. And sometimes the inhibition of reaction is also done by disabling or removing the enzyme necessary for the reaction. And this can be done by in a couple of different ways. One can use enzyme inhibitors, which are chemicals that bind to the enzyme and prevent it from performing function as a catalyst. Or one can genetically alter the organism under study to remove or impair its ability to produce the enzyme. This is called knockout experiment. The same technique can also be used to determine which reactions are catalyzed by which enzymes is the first place, and hence to discover the structural third enzymatic part of the tripartite metabolic network. So the construction of a complete or partial picture of metabolic network, it involves different types of data from different pathways, and almost certainly derived from experiment performed by different experimenters using different techniques. So now there are number of public databases of metabolic pathway data from one can draw to assemble networks to best knowing and metaseq. And assembling the network itself is a non-trivial task because data are drawn from many sources, careful checking against the experimental literature, that is called curation, is necessary to ensure consistent and reliable input to the process, and missing steps in the metabolic pathway often be filled by guesswork based on biochemistry and knowledge of the genetics. And therefore, there are a lot of some computer softwares that are developed to reconstruct networks from the metabolic data in an automated fashion, but the quality of the networks they create are thought to be poorer than networks created by knowledgeable human scientists, although computers are much faster. So these are the things, basic idea about metabolic network. I will come for this metabolic modeling also later on. Okay, when we will study in detail how to do the metabolic modeling. And then another type of network is a protein-protein interaction network. Now, if you see the protein-protein interaction network, they have a protein interact with one another and with other biomolecules, and the interactions are not purely chemical. Sometimes they interact chemically with other molecules, so they exchange a small subgroup, and such as exchange of phosphate group in the process called phosphorylation. The primary mode of protein-protein interaction of protein with other protein is physical, and the complicated folded shapes interlocking to create called protein complexes, but without exchange particles or subunits that define the chemical reaction. Therefore, a set of all protein-protein interaction forms a protein-protein interaction network in which the vertices are proteins and two vertices are connected by an undirected edge if the corresponding proteins interact. Although this representation of network is one commonly used, it omits much more useful information about the interaction. When interactions involve three or more proteins, they are represented by multiple edges, and there is no way to tell from the network itself that such edge is an aspect of same interaction. So these problems are tight representation of the network, as we told for metabolic network, and there are a number of experimental techniques to prove the interaction between the protein. So one of the most reliable technique is a co-immunoprecipitation. Co-immunoprecipitation is a technique for extracting a single protein species from a sample containing more than one, and this is the interaction between antibody and antigen. And another technique is the by co-immunoprecipitation we can identify the protein interaction, number one. Number two is that co-immunoprecipitation is a practical approach of reconstructing entire interaction network. So when you are talking about the whole network, it is very difficult for you to do for all. Therefore, if appropriate antibody is created by processes that will take even a longer time, and creation of single antibody involves weeks and months of work and cost of considerable amount of money. As a result, the large-scale study of protein-protein interaction network didn't really take off until the adoption of 1992,000 of so-called high-throughput methods of discovering interaction. So now we have a high-throughput method, and now the oldest and best established high-throughput method is the two-hybrid screen. Okay? And here you know that this method relies on the action of a specialized protein called transcription factor. So if present with the cell, it turns on the production of another protein known as reporter. The presence of reporter can be detected by experimented by any of the number of relatively simple means. The idea of two hybrid screen is to arrange things so that transcription factor is created when two proteins of interest interact, and thereby turning on them and tells us the interaction has taken place. The two hybrid screen relies on the fact that transcription factors are typically composed of two distinct parts, so-called binding domain and activation domain. Therefore, the most transcription factors don't require the binding and activation domain to be actually attached to one another for transcription factor to work. If they are nearly close enough proximity, production of reporter will be active. So in case of two hybrid screen within a cell, usually each cell is persuaded to produce two proteins of interest, each with one of the domains of transcription factor attached to it. This is done by introducing a plasmid into the cell, fragment of DNA that code for the protein and domain. If the two proteins are questioned, interact and form a complex, the two domains of the transcription factor will be brought together and with luck will activate production of reporter. In a typical two-hybrid experiment, the protein attached to the binding domain of the transcription factor is a known protein called the bait protein, whose interactions the experimenter wants to prove. plasmid coding for a large number of other proteins like prey attached to the copies of activation domain are created, and therefore a library of possible interaction target for the prey. So the plasmid and library for the prey are then introduced into culture of each cell, and concentration of prey carefully calibrated so that one prey plasmid enters each cell in most of the cases. So two hybrid technique has two important advantages over other methods like co-immunosuppression. First, that one can employ a large library of prey and test for interaction with many proteins in a single experiment. And second, the method is substantially cheaper and faster than co-immunoprecipitation for interaction detected. The co-immunoprecipitation requires 1 to 10 or create antibodies for every protein tested. The two hybrid screen requires only the creation of DNA plasmid and their later sequence analysis, both relatively simple operation for experimental armed with the machinery of modern technology. One disadvantage of two hybrid screen is that presence of two domains of transcription factor attached to the bait and prey protein can get in the way of their interacting with one another and prevent the formation of the protein complex, meaning that some legitimate protein-protein interactions will not take place of the experiment. Therefore, the principal disadvantage of the method is that it is simply unreliable. It produces high rates of false positives, apparent interaction between the protein that in fact don't interact, and there will also be a false negative result that there is a failure to detect true interaction. By some estimate, the rate of false positive may be high as 50%, meaning that only half of the detected by the method are not here. This has not stopped a number of researchers performing an analysis using interaction networks reconstructed from two hybrid screen data, but the results should be viewed with caution. So every technique has its own limitations. Similarly, each two hybrid technique also is having its own limitation. So it is always desirable to test your hypothesis using multiple techniques. And there is also another technique called affinity purification method. This is similar to co-immunoprecipitation, but avoid the need to develop antibodies for each protein probe. In affinity purification method, a protein of interest is tagged by adding a portion of another protein to it, typically by introducing a plasmid that codes for the protein plus the tag, in a manner similar to introduction of plasmids into hybrid screen. The protein is given the opportunity to interact with suitable library of other protein, and a solution containing resulting protein complexes passed over a surface to which are attached antibodies that bind to the tag. As a result, the tag attached protein and its interaction partners, they are bound to the surface while the rest of the solution is washed away. Then by co-immunoprecipitation, the resulting solution is washed away. Then, so the advantage of this method that it requires only a single antibody that binds to the known tag, and same tag antibody in different experiment to bind the different protein. Thus, with the two hybrid screen, one need to generate new plasmids for each experiment, which is relatively easy as opposed to generating new antibodies. So you can use this this protein-protein interaction, and this diagram shows the protein-protein interaction network of yeast. Now there is also a network called genetic regulatory network. Here the small molecule aided by biological organism like sugar and fat, they are manufactured in the cell by cell reactions of metabolism. Proteins, however, which are other molecules, are manufactured by a different manner, as you know. So proteins are formed from amino acids. And then there is also a process called folding of the protein that is important. So now I'm not going in biochemical properties of proteins and synthesis of protein, but I will go directly to the gene regulatory network. So these transcription factors, they are themselves produced by transcription from gene. So the protein encoded in a given gene can act as a transcription factor, promoting or inhibiting production of one or more other protein, which themselves can act as a transcription factors for further proteins and so forth. The complete set of such interactions form a genetic regulatory network. So whatever interaction is going on at the level of as a transcription factor, this whole set of interactions is called genetic regulatory network. And the vertices of the networks are here proteins or equivalent to the genes that code for them, and directed A's from the gene A to gene B indicate that A regulates the expression of B. A slightly more sophisticated technique is that promoting and enabling transcription factors. So the experimental determination of this regulatory gene genetic regulatory network, it involves the identification of transcription factors and the genes they regulate. The process has several steps. First, we have to confirm that a given candidate protein bind to a DNA roughly in the region of the gene of interest. The commonest technique is to operative mobility shift assay. In this technique, one creates a strand of DNA containing the sequence to be tested and mixes them to solution with candidate protein. If the two proteins bind, then the combined DNA protein complex. If the two protein and DNA binds, then this combined DNA protein complex can be detected by gel electrophoresis, a technique in which one measures the speed of migration of electrically charged molecules or complexes through agarose or polyimide gel in an imposed electric field. In the present case, the binding of the DNA and protein, it hinders the molecular motion of the resulting complex with the gel, and then it measures the reducing the speed with compared to unbound grain strand. Typically, one side by side with one protein. Now, 1 minute, let me check. We were supposed to go on a meeting today. Yeah. There is a meeting. Okay. So I will just stop here because there is a meeting at 11:30, so I have to go for the meeting. So I think you have talked, and we'll continue this in future classes. And this, whatever I'm discussing is from the book named Networks and Introduction by M.E.J. Newman. Okay, this is Oxford University Press book. I think now you know how to download the different books, so you just download this book. Okay. You have any queries before I close? So tomorrow we will continue with this. Today who is present? Amal is present. Arnav is present. Pablo is present. Chandan is present. Chandini is present. Vidya is present. Hema is present. Matangi is present. Megha is present. Rasi is present. Yeshi is present. Sadia is present. Satish is present. Subhash is present. Jayavanand is present. Today Vishal is absent, I think. Yeah, he's not this. Okay. So tomorrow we'll meet at same time, around 10:45, and we'll continue with whatever I have discussed. Okay. So
Thank you, sir.
you download the book and go through the book. It's a very nice book. Okay. It will help you in understanding.
Can you please start that also?
Network. I told you, no? Newman. Just to see network and introduction. Newman. 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 Okay.
Okay, sir.
Yeah. Okay. Thank you. So we'll meet tomorrow.
Thanks, sir.
Thank you.
Thank you, sir.